Well, it's all right.
I have no idea why the screen appears green, but hopefully you'll be able to see the slides. So, quick reminder, for some of you anyway, there's an exam tonight. It's in 11:27, so don't show up here. It's a common mistake that people make coming back to where, yes, question?
So is this test only an hour now? So if we schedule a few?
Oh, I'm sorry, that's a typo on my part. You have 2 hours. See, and you don't have to be there at 5:30 if you don't want to. That should say 5:30 to 7:30. Thank you for noticing that. So for maybe 10 or 12 of you that will show up tonight, that's this will be the exam. Otherwise, let me know, and you can let me know anytime. You can change your, if you're signed up for tomorrow and suddenly you realize tomorrow it's like, damn, I'm not ready. Just send me, just don't come take the exam. Send me an email and reschedule it. So as little stress as possible. Okay. So we left off talking about bacteria's adaptations to extreme temperatures. So bacteria that live at low temperature, we kind of covered some of their adaptations at the molecular level. But now let's talk about adaptations of the hyperthermophiles, which are mostly archaea, and the thermophiles, which are both archaea and bacteria. So I was kind of going through this a little bit in a rush at the end of the last lecture. I'm sorry about that. So the enzymes of these thermophilic and hyperthermophilic microbes actually work best at temperatures that would denature enzymes in your cells or my cells, or E. coli or staph or any of the bacteria that you've grown to know and love. But what's really interesting, so when people began to be able to sequence pretty reasonably genomes, I think a lot of us were expecting that when you looked at the homologue of, say, DNA polymerase from a hyperthermophilic organism, that you were going to see wild differences between the enzyme at the genetic level between a hyperthermophile and, say, a mesophile. And surprisingly, the differences are profound, but they're not as striking in terms of the number of sequence differences that they're there. So it was really kind of surprising. And a lot of the differences that occur, even though they're few in number, are pretty profound in terms of their effect on the stability of the enzymes. And so they're exactly what you would predict. There are more substitutions of ionic amino acids, which can lead to salt bridges. So these, you can imagine, would give an enzyme more stability in a high thermal environment to keep them from denaturing. The other thing that's pretty striking is there is an increased amount of hydrophobic amino acids. Now, in the last lecture, when I mentioned that psychrophilic bacteria that live at cold temperatures, when I talked about their molecular adaptations, one of the bullet points was that there was a decreased amount of hydrophobic amino acids. So this makes sense in terms of these two types of organisms having exactly opposite adaptations in terms of hydrophobic amino acids. But I think that the mechanism is easier to consider when you want to think about why is it that an increased content of hydrophobic amino acids would help a thermophilic organism's enzyme survive. If you think about one of the major forces that causes proteins to fold into their secondary and tertiary structures, one of those is to protect hydrophobic, water-hating amino acids from being on the exterior surface of the protein, exposed to the aqueous environment. So if you look at the amino acids in a protein that tend to be in the internal portion, the area of the protein that maybe water is excluded from, those tend to be hydrophobic amino acids. So what thermophilic bacteria and hyperthermophilic archaea have done is that when you look at the homologous protein It could be DNA polymerase, RNA polymerase, the things that all bacteria have to have, regardless of the temperature that they live at. If you think of these bacteria as having an increased number of hydrophobic amino acids, that adds to the stability of the protein, because imagine that as that secondary and tertiary structure begins to unfold at high temperature, if there's increased hydrophobic amino acids buried in the middle of that protein, that's an increased force trying to work in the opposite direction, trying to stay away from water. So the increased hydrophobicity of those amino acids adds to the stability because it tends to make the protein resist thermal denaturation. As the protein tries to denature, those hydrophobic amino acids become exposed to water, and so they hate that. And so the conformation is there's a force acting against that. So that's why I said I think it's a little easier conceptually to think about this adaptation when we're talking about thermophilic bacteria and hyperthermophilic archaea. So besides protein stability and membrane stability, which we've talked about a little bit with the psychrophiles, there are other ways to die at high temperature. So what other adaptations do microbes have to live at really high temperatures? So one is, again, it makes sense in terms of the membrane adaptations. We talked about the fact that psychrophilic or cold-living bacteria, loving bacteria, have increased unsaturated amino acids. So they put kinks in their fatty acid chains. It's the opposite for thermophilic bacteria. This way, in a high thermal environment, the van der Waals forces are greater because you can pack those phospholipids closer together. So the fatty acids are going to interact with one another more. So thermally, that membrane is not as likely to dissociate at high temperatures. And then, of course, we had talked about this with the archaea, that in hyperthermophilic archaea, oftentimes the membrane is not a lipid bilayer, but instead it's a lipid monolayer. So the two leaflets of the membrane can't pull apart due to high temperature. They're covalently bound to one another. So we see these lipid monolayers almost exclusively in the hyperthermophilic archaea. And then, of course, there are proteins that help keep the DNA stable. So DNA, as you all know, can be melted or denatured into single-stranded DNA if the temperature rises. But one of the ways that especially archaea have evolved to keep their DNA intact is to use DNA binding proteins and DNA binding solutes, such as potassium cyclic 2,3-diphosphoglycerate. I don't know what the hell it does, but it is a chemical that, for reasons that are beyond my ability to comprehend, makes DNA less likely to denature. So it's something that's produced in these organisms, but not in most other organisms. And then there are DNA binding proteins. Remember that, although we're not talking about histones now, histones are actually invented phylogenetically. The same histones that you and I use to package our chromatin are used or are invented in archaea. But they also have things like this SAC7D, which is a heat-stable DNA binding protein that tends to keep DNA from melting. And then this is the only place that you find this in nature, is reverse DNA gyrase. So we talked about DNA gyrase in the first portion of the class, helping bacteria to package their DNA to form negative supercoils. The only place that we see positive supercoils is in these hyperthermophilic archaea, where they actually have an enzyme appropriately enough named reverse DNA gyrase, because it forms positive supercoils, which, again, I can't conceptualize exactly why supercoiled DNA, in a positive sense, is more stable at high temperature, but it is. So this is another adaptation that we see to high temperatures. Now, I mentioned when I put up a table earlier, the only bacterium that I was really interested in you knowing anything about and remembering the name of and anything about the biology is a thermophilic bacterium called Thermus aquaticus. So this picture is probably not entirely appropriate. This is a volcanic feature that probably has temperatures, since it's boiling and steaming like this, probably over 100 degrees. Thermus aquaticus, the bacterium that we're talking about now, actually would probably die at something quite that hot. But But as that water in these thermal features in places like Yellowstone National Park tends to come away from the heat source, it tends to cool. And so you find a gradient in temperatures in these thermal features in places like Lassen Volcanic and Yellowstone National Park. And so at some point there is an ecosystem in that water system where the water is around 72 degrees pretty consistently. And that's where Thermus aquaticus lives. It was actually isolated from there. So it's a pretty boring looking bacteria, like most bacteria are, just a rod-shaped bacterium. But actually it's the enzyme, the DNA polymerase from Thermus aquaticus that has really revolutionized molecular biology research in all areas of research. So you may have heard of Taq polymerase. So a lot of times when you see three-letter abbreviations for enzymes, so we'll talk about restriction enzymes later, like BAMH1, EcoR1, those letters are actually not just randomly chosen. They usually reflect the genus and the species of the bacterium from which they come. So T from Taq polymerase comes from Thermus, AQ, the first two letters from the species name. So Taq polymerase is used in the polymerase chain reaction, or PCR. And so I know probably many of you, most of you perhaps, understand this process and have probably done it, but this is just a little narrated video to keep you from having to listen to me the whole time, but also to explain how Thermus aquaticus accomplishes polymerase chain reaction. Well, I think it was working this morning. We're going to skip that for now. I'll put that video on Blackboard so you can see it. But basically, in essence, what's going to happen is that Taq polymerase, because it's thermostable, can be heated to very high temperatures without denaturing. And so what that allows us to do is to amplify large amounts of specific DNA sequences for the purpose of cloning, for the purpose of sequencing. And so this has really made so many experiments that were impossible even when I began graduate school, now just routine. Actually, I can remember the first time, when the first paper came out on PCR, I can remember my best friend from another lab, Lisa, telling me at lunchtime one time, "Did you see this paper about this chain reaction they could do to amplify DNA?" And I hadn't read the paper yet, and I thought, oh, it's probably not very important. And it ends up being something that's just changed everyone's careers. Okay, so now I want to move on and talk about what's one of my favorite topics in microbiology, and that's transcription. So my personal interest is more about gene expression and control of gene expression. So I like this. But in terms of comparing transcription, we won't go too much into translation. I've taken that section out to shorten the class a little bit. We're going to talk mostly about comparative transcription between prokaryotes and eukaryotes. So in prokaryotic cells, bacteria and archaea collectively, oftentimes what you see is that the DNA, at the DNA level, you can have adjacent genes that can be transcribed into messenger RNA as a single messenger RNA. So 2 or 3 or 10 or 20 different genes that are adjacent to one another can be co-transcribed into a single piece of messenger RNA, and then that single piece of messenger RNA can be translated into individual proteins. So this is polycistronic DNA. This is an operon. This particular case is a very simple operon. It's only 2 genes. But bacteria can have 17 to 20 or 30 different genes in one particular operon. So one piece of messenger RNA codes for multiple proteins. Compare that to transcription in a eukaryote. This very oversimplified cartoon demonstrates that in eukaryotic cells, virtually every gene is monocistronic. In other words, there's one transcript for every gene, for every protein to be produced. So operons, as they exist in bacteria and archaea, largely don't, if ever, exist in eukaryotic organisms. There are probably some rare exceptions to that. But you typically have one gene, one RNA, and then that RNA will be spliced, will be processed in most cases. So introns are removed and exons are spliced together. This type of processing virtually never occurs in bacteria, and there are very few examples of such processing that occur in archaea. So RNA splicing and religation to get rid of intronic sequences is pretty much unheard of in the prokaryotic world. But in the eukaryotic world, it's probably more the rule rather than the exception. And so you have a mature piece of messenger RNA encoding a single protein. And then, of course, remember that eukaryotes have nuclei, whereas prokaryotes don't. So this entire process, the transcription and the splicing, is occurring in the nucleus. And then the messenger RNA is transported into the cytoplasm to be translated into a single protein, so monocistronic RNA. That's the other really interesting and impactful difference between transcription and translation in a eukaryotic cell versus a prokaryotic cell. In a eukaryotic cell, the generation of this protein is by translation only occurs in the cytoplasm. The transcription occurs in a completely different compartment in the nucleus. In a bacterium, because there is no nucleus, transcription and translation occur in the same compartment. In fact, they occur at the same time. As this piece of messenger RNA is being transcribed but is not completed in its transcription, ribosomes will have already begun the translation of protein A. So protein A may be completely translated from this messenger RNA before this piece of messenger RNA is even completely transcribed. So transcription and translation are coupled in prokaryotic cells. This has a profound difference on the biology of bacteria. So one of the things that I'm going to want you to appreciate is not simply to memorize this seemingly trivial fact that transcription and translation are coupled in prokaryotes but not eukaryotes, but we're going to go into why that fact has very profound differences, or makes for very profound differences in the biology of bacteria and archaea. Now, the enzyme that that carries out transcription is RNA polymerase. Very, very simple naming scheme. Everyone realizes this. But if you look at a table and compare the RNA polymerases between prokaryotes, bacteria on the left, archaea here in the second column, and eukaryotes, one of the striking things that you see is that the archaeal RNA polymerase is much more closely related to ours in eukaryotes than it is to bacteria. So despite the fact that archaea are considered prokaryotes, they are prokaryotes, they're actually more closely related to eukaryotes than they are to bacteria. So, this term prokaryote versus eukaryote is really a man-made distinction. It doesn't, it really has more to do with the lack of a nucleus, the lack of organelles. But as you'll see as we talk more and more in this class, there are times where you'll be surprised to find that these bacteria-looking organisms, archaea, they were actually considered bacteria even when I was in graduate school in the 1980s, are actually much more closely related to us. In bacteria, the RNA polymerase is relatively simple, or at least relatively compared to eukaryotes. It has a beta and a beta prime subunit. There's a typo on this. One of these should be prime. That's a minor point. Then there are 2 alpha subunits and an omega subunit, and they've left out an important transcriptional factor, which we'll come to. But you can see that here are only 5 protein subunits to make up the RNA polymerase, which does all of the RNA synthesis within that bacterium. In archaea, there is a single RNA polymerase that does everything in there, transcribes everything, anything that needs to be made into RNA is made by a single RNA polymerase in archaea. But if you look at the subunits that make up the archaeal RNA polymerase, it's much more complex. There are extra subunits for which there are no homologs in bacteria. But these additional subunits do have homologs when you look at the 3 RNA polymerases that eukaryotic cells have. So clearly, archaea's transcription is much more like eukaryotes than it is like bacteria. If you look at the 3 different RNA polymerases of eukaryotic cells, you can see not only are they complex individually, so multiple subunits to make up eukaryotic RNA polymerase II. We're not even talking about the transcription factors that bind elsewhere on enhancers upstream and downstream. This is a relatively, or not a relatively, this is a complex multi-subunit enzyme. But there are 3 different ones. So eukaryotes have actually, in the course of their expansion of their genome and the gene families that they can make, have actually subdivided the work in terms of making RNA in a cell between 3 different, completely different RNA polymerases. RNA polymerase II generates all of the ribosomal RNA of the cell, and that's exclusively its job. There is a completely different enzyme, RNA polymerase III, which makes all of the messenger RNA. So those pieces of RNA that need to be translated into proteins are made exclusively by this RNA polymerase. And transfer RNA synthesis is handled by yet a third, completely different RNA polymerase. So highly complex, highly involved transcription in eukaryotic cells, slightly less complicated in archaea, and simpler in bacteria. Yet, as you're going to see, bacteria need to do the same division of labor that eukaryotes do, and they've just evolved to do the same thing by a completely different mechanism. So they are simpler, they are less complex in terms of their subunit structure, but they also have the ability to specialize as well. So this is just a cartoon showing RNA polymerase, one of the RNA polymerases of eukaryotic cells. So this mouse-looking thing here is RNA polymerase. It interacts with a lot of different transcription factors. It binds to a particular spot on the DNA, this Tata box. But then you have all of these upstream sequences, which can be hundreds, thousands of base pairs upstream or downstream, in fact, in the coding region of the gene, that are going to bind transacting factors, other transcription factors, that have the ability to loop back and interact with this RNA polymerase to either facilitate transcription initiation or to inhibit transcription initiation. So really highly complex in eukaryotes. And there are 3 of these, 3 different of these enzymes to accomplish this. When we look at bacteria, thankfully, it's much less complicated. Glad I don't have to teach eukaryotic transcription and to go through all of those different protein subunits. Forget about this red subunit here. This is actually a transcription factor. I stole this from something that wasn't really related, but I like this picture. So this portion, or let's look at the bottom portion first, because here you can see the RNA polymerase pretty much in exclusion. Again, forget about this blue molecule here on the left at the bottom. The RNA polymerase of all bacteria have a beta and beta prime subunit, so they're not identical to one another. In some bacteria, they're actually fused into one gigantic protein. The beta subunit of RNA polymerase is almost always the largest gene in any bacterial genome. You can spot it looking through the genomes quite easily just because it's so damn big. And in some bacteria, it's actually fused with the beta prime, so it's enormous gene. But the beta-beta prime subunits actually will also be attached non-covalently to the alpha subunit, so 2 alpha subunits. So what this picture is showing is the carboxy terminal domain, that's what CTD stands for, and then the amino or N-terminal domain. So this is all one protein, 2 copies, 2 identical copies of alpha, with the N-terminal domain interacting with the RNA polymerase beta subunits. The alpha subunits the carboxy terminal domains of the alpha subunits are usually used to interact with transcription factors. So that's what's shown here in the bottom is that the carboxy terminal domain can interact with some transcription factor or some number of transcription factors which can bind to DNA sequences upstream of where the RNA polymerase is binding. The other things to notice in this cartoon and to remember are the means by which this polymerase, this RNA polymerase, binds to a specific sequence. So there's one subunit that wasn't listed in the previous table, and it's shown here as this brown subunit, sigma. Sigma is the subunit, so sigma, or S, stands for specificity. So you can always remember that sigma, or the S subunit, is what allows for the RNA polymerase to bind to specific DNA sequences. So shown here on both the upper and lower cartoons, you can see these two kind of golden boxes at the DNA level. Those are collectively the promoter sequence. As it turns out, the one furthest to the right is referred to as a -10 box or -10 element. The one behind that is the -35 element. That minus has to do with the start site of transcription. So the +1 here indicates that is the DNA sequence that will initiate, that will be the first nucleotide of the messenger RNA that's made by this RNA polymerase once transcription is initiated. But that sigma factor is absolutely crucial for binding to this -10 and -35 promoter sequence. It's what makes the transcription be specific for a particular gene. Now, the reason that that previous table left the sigma factor off is that this sigma factor has absolutely nothing to do with the synthesis of RNA. It is a subunit that comes and goes from this RNA polymerase, and I'm going to cover this again. But this sigma factor's only role is to find that promoter, that -10 and -35 sequence, and to initiate transcription. And then it's useless. It's discarded. As a matter of fact, it would probably work against the enzyme because it actually would tend to stick it to this -10 and -35, make it adhere to that point. So that's why that table didn't list sigma. It actually treats sigma as a transcription factor, which in fact is probably a pretty good way to think about sigma. Rather than think of it as a subunit of RNA polymerase, you can easily think of it as a transcription factor, something that facilitates transcription. Now, kind of staying with that, we're going to come back and look at the subunits, especially the sigma subunit and its functioning in a little bit. But let's talk about this transcription in bacteria and how it differs from archaea. So again, now we're looking at the DNA. The polymerase is not here. Here is collectively the two sequences that are typically referred to as the promoter. So -10, -35. And again, -10 simply means that the center nucleotide in that hexamer. So the -10 sequence is typically 6 nucleotides in length. The center nucleotide of that is usually 10 base pairs, almost exactly upstream from where the first nucleotide of the messenger RNA is. That's why it's called -10. What -35 behind that, and you'll find another 6 base pair sequence, another hexameric sequence to which sigma binds. So both the -10 and -35 tend to be hexameric sequences, and they're somewhat conserved. We'll talk more about that. But so this is where the RNA polymerase would bind. This is the promoter. Now, I don't like the terminology that this slide used. They talk about this next sequence here between these two hash marks as a leader sequence. I prefer the term of 5' untranslated region. So 5' untranslated region of messenger RNA is very descriptive. It's the 5' end. It's not translated into a protein. So it's a 5' untranslated region, sometimes problematically for me called a leader sequence. And so if you look at here, just below that, here is the resulting messenger RNA that would be created. RNA polymerase would bind to the DNA at this promoter sequence, and it would begin transcription at the +1 sequence. So it would make its first nucleotide, usually a purine nucleotide, so either guanine or adenine is usually the first nucleotide in messenger RNA. That's kind of a trivial point. And then it transcribes from left to right, making a copy of the antisense, or the bottom strand here. So here you have the messenger RNA. Notice that the promoter region is not part of the messenger RNA. So the region that bound to that RNA polymerase does not become part of the RNA transcript. It's the same thing in eukaryotic cells as well. But so here's the 5' end with a G and A. There is a really important sequence that's encoded in the DNA, just a few nucleotides upstream of the start codon. So this hash mark on the red line, the second hash mark downstream of the +1 site, is meant to indicate the start codon of that protein that's encoded at that DNA level. So when this becomes transcribed, you'll see a sequence here about 8 to 10 base pairs upstream of that start codon, which is called a Shine-Dalgarno sequence. You can interchangeably use these two terms, Shine-Dalgarno and ribosome binding site. Ribosome binding site is actually a more informative term. It's easier to remember what a ribosome binding site does. It binds a ribosome. But for historical purposes, because the people who worked this system out, it was originally named in honor of Shine and Dalgarno. So it's still very frequently referred to as a Shine-Dalgarno or just an SD site. But in reality, you can just call it a ribosome binding site. Ribosomes don't bind to DNA. So this sequence, this Shine-Dalgarno sequence, although it's only labeled here on the RNA, it occurs as a DNA sequence on the double-stranded DNA, but ribosomes never bind there. Ribosomes only bind single-stranded RNA. So they'll only bind here and begin translation at this start codon. Usually a methionine codon, AUG, but bacteria are unusual in that whereas in eukaryotic cells, AUG is, if not always, pretty close to always the start codon for all eukaryotic proteins. Bacteria can also use GTG, or what would be GUG, or UUG, which would normally encode a different amino acid. Yes, question?
So on the bottom, is there any direction?
Yeah, actually, I never noticed that before. Yes, the ribosome would bind onto the Shine-Dalgarno sequence and translate in this. But I think what they're really meaning to say is that this DNA was transcribed from left to right. But then, yeah, you're right. This should actually say translation to be technically correct. Good catch. Okay, so now that I've talked, I think I've gotten ahead of my bullets. I mentioned the 3 RNA polymerases in eukaryotic cells, and the most common bacterial promoter consisting of the 2 hexamers at -10 and -35. So we've gone through the fact that it begins with a purine and RNA, yep. The 5' untranslated region is absolutely crucial in eukaryotic, in prokaryotic messenger RNA because there has to be some type of an untranslated region because that's where the ribosomes bind. 5' untranslated regions are much more extensive in eukaryotic cells. So the 5' untranslated region of eukaryotic genes can be hundreds or thousands of base pairs long. Typically in bacteria, 100 base pairs would be a really long 5' untranslated region. But the reason why they're very important is because they have to encode the Shine-Dalgarno sequence for the ribosome to bind. So the way that the RNA polymerase initiates transcription, once it has locked and loaded onto that promoter sequence, and remember it loaded onto that promoter sequence using the sigma factor or specificity factor. So in this particular cartoon, you can actually see the sequences of the 2 hexameric sequences. So here's the -10 sequence, TAT, GTT. This is not a perfectly consensus sequence, but it's close to what the consensus sequence for a -10 is. And here is a -35 sequence, TTT-ACA. So those are the 2 sequences that sigma would recognize, so directing the RNA polymerase onto this molecule. And then what happens is what's known as the formation of the open complex. This is a transcription bubble. So the RNA polymerase will now melt the DNA locally into a bubble here of about 12 or 15 nucleotides at any one time, and has begun to put nucleotides on complementary to that antisense or bottom strand. So you see here it's putting on a GA, and it will just continue in that particular direction. So that's what the RNA polymerase does to initiate transcription. It does this local melting. Now, if the RNA polymerase remains the way it is with a sigma factor, as well as beta, beta prime, the alphas, and there's a weird little subunit called omega that I don't know much about. I don't think anyone knows exactly what omega does. But if the RNA polymerase maintained that stoichiometry, that construction, it would really kind of be stuck there because the sigma factor really likes this -10 and -35. And so what happens is that it has to shed the sigma factor to allow what's known as promoter escape. The RNA polymerase has to escape that promoter. It might be kind of glued there a little bit by the sigma factor. So sigma is kicked off, Once this open complex has been made, and the first few nucleotides have been synthesized by RNA polymerase, sigma is no longer needed, it's jettisoned. So sigma can be recycled. Sigma will just bind to another RNA polymerase that doesn't have a sigma, and hopefully bind to that same promoter again to start making the next transcript. But in the meantime, the RNA polymerase minus sigma now is moving downstream and making messenger RNA as it goes along. So sigma is absolutely, completely unnecessary for the synthesis of messenger RNA. Its only job is to begin the process. So this is an even simpler way to look at this. Here is one form of the RNA polymerase called the core enzyme. The core enzyme of beta, beta prime, and 2 alphas, and the omega, not shown here, is the only thing necessary for RNA synthesis. So this is the actual machine that makes messenger RNA, but by itself, it has no way to find where it needs to start the messenger RNA. So when a sigma factor binds to this, now the sigma factor can add that specificity. This is what's referred to as the holoenzyme, RNA polymerase holoenzyme. It can find the promoter, initiate transcription, kick off sigma, and then just continue trucking on down the road, making RNA as it goes. Now, this is the point where, so remember I talked about the fact that eukaryotic cells have evolved this very beautifully complex system of using 3 different RNA polymerases to do 3 different jobs. Bacteria have evolved the ability to make sure that they have RNA polymerases to do very different individual jobs. But rather than make a completely new RNA polymerase, what bacteria do is that they substitute different sigma factors in. So the sigma factor here might bind to a -10, -35 sequence that's TAT-AAT at the -10 sequence, and that might transcribe one set of genes. In fact, it would. Probably, the majority of the housekeeping genes are done by one particular sigma factor. But under different conditions, as the bacterium changes its position, as it changes parts of its growth cycle, there are different genes that are required to be transcribed. And so those genes will require will have a different promoter, and so a different sigma factor will bind. So E. coli has 7 different sigma factors, but remember, E. coli is what's referred to as a facultative host-associated organism. It's a facultative anaerobe. We talked about that already. But E. coli is one of these bacteria that has evolved to live in association with the host in our colons. But at some frequency, it survives in the environment in order to find another host. So it does have some kind of an environmental portion of its life cycle. So it still has enough different environments that it encounters, both in the colon and in the environment, that it has evolved the ability to transcribe all of its genes using one of 7 different sigma factors. They will have a sigma factor that is necessary if the bacterium finds itself at high temperature, a heat shock sigma factor. There is a sigma factor that E. coli will use exclusively during the stationary phase. So genes that need to be transcribed during that stressful part of the bacteria's growth curve are transcribed with a different sigma factor. Bacillus subtilis, the bacteria that you did, the soil bacteria you did spore samples on, is a soil bacterium. So it encounters even more different types of environments. So it has an even more complex arrangement of genes to be transcribed. It has 17 different sigma factors, any one of which can substitute into the RNA polymerase core enzyme to make a holoenzyme that has a completely different specificity. So a different suite of genes will be transcribed under each of these 17 different RNA polymerases. I don't know why I duplicated that RNA polymerase from the eukaryotic cell, so there must have been a reason. It was probably to contrast it, perhaps, with this model, of bacterial RNA polymerase bound to DNA. So the sigma factor here is shown in red. The DNA is obviously this helix near the top. And you can see that the sigma subunit here has 2 different domains that interact with the DNA. It has one domain that's interacting with the -10 element. So extended -10 elements are just 9 base pairs in length as opposed to the typical 6. This paper had something to do with that. But it was a nice picture, so I stole it. But it shows the -10 sequence interacting with one portion of sigma. Another portion of that same protein, you can't see the connection between this domain 3 and domain 4, but they're part of the same protein. Domain 4 is interacting with the -35 element. And here you have two domains on the carboxy terminal portion of the alpha subunit that are interacting with DNA sequences upstream. So you'll notice that there's a bend in the DNA. This is actually something that we study in our lab, is how RNA polymerase in Helicobacter pylori has the ability to bend DNA in order to interact with upstream elements at the DNA. But then the rest of this molecule is actually what's necessary for synthesis. So this is bound to a promoter. Here would be where transcription is going to start. It hasn't started yet, so sigma is here. But as soon as this area melts slightly, the transcription will begin, sigma will be shed, and promoter escape will occur, and the RNA polymerase core enzyme, not the holoenzyme, the core enzyme will continue down to the right, making messenger RNA. And I decided I would repeat that picture again for some reason, just to show you again, I guess, that sigma is binding to the -10 and -35. These are those carboxy terminal domains of alpha that can interact, as in that previous picture, with DNA, but they can also interact with transcription factors that are bound upstream. Now, I mentioned that E. coli has 7 different sigma factors, and these sigma factors are obviously going to bind to different promoters that would control the transcription of different genes. Now, there is one particular sigma factor that we know most about, sigma 70. Sigma 70 in E. coli, the 70 stands for its size. It's 70,000 Daltons in size. It does not stand for the fact that about 70% of all of the E. coli genes are transcribed using sigma 70. But this is the general housekeeping sigma. So as E. coli is growing in logarithmic phase, growing very well in the laboratory, most of its RNA polymerase is going to be bound to sigma 70. Sigma 70 is going to bind to various promoters of slightly different sequences. But there is a consensus sequence for sigma 70 that you do not need to memorize. Please don't memorize something like this. But the sigma sequence minus sigma 70 consensus sequence of -10 is this TAT-AAT. The consensus -35 is TTG-ACA. Now, that's the best sequence that sigma 70 can bind, but it's not the only sequence that it will bind. If you look here in this cluster W lineup of sequences from E. coli promoters, these are all -10 and -35 sequences of different genes in E. coli, each of which is bound by RNA polymerase with sigma 70. But you see that the sequences are not exactly the same. But they're in the same locations relative to the -10 to the -35 and relative to the start of transcription. The advantage that the bacteria has by slightly varying the promoter sequence away from the consensus is that it affects the promoter strength. Not all promoters are created equally. And some promoters are in front of genes that need to be transcribed at very high levels. And some are in front of genes that need to be transcribed at very minute levels. And one of the ways that bacteria have evolved to make sure that genes that really need a robust transcription is that they'll have -10 and -35 sequences that are very close, if not exact matches to this consensus sequence. Because sigma 70 sees that and it just binds on and starts transcription like there's no tomorrow. Whereas other promoters that are in front of genes that just vanishingly small amounts of the protein or transcription are necessary, they'll have sequences that differ. So here on the bottom, this is only a 4 out of 7 match at the -10 sequence, or -35, but a perfect match at the -10. That's going to have a different rate of transcription initiation than promoters up here where you have a 3 base pair match and a 2 base pair mismatch in there. So promoter strength is dictated by how close these -10 and -35 sequences match. But again, you have, as well as having different promoter sequences for sigma 70, you have completely different promoter sequences to recognize different sigma factors. So these are the main sigma factors, the best studied sigma factors in E. coli. And you can see that, for instance, sigma 32, again, 32 reflects the size of this protein. This is a protein that's very important for transcribing genes that the bacterium needs to survive if it finds itself at a high temperature, so heat shock. There are genes that help a protein survive heat shock because proteins tend to denature as temperatures rise. And so the bacterium needs to produce chaperone proteins, which help refold proteins. So those genes would have in front of them a promoter sequence that was different than sigma 70. They would have sequences that look like this at minus 10 and this at minus 35. So these wouldn't be transcribed under normal conditions. They wouldn't be transcribed until conditions were such that sigma 32 was now the predominant sigma factor. So the bacteria has a means that we'll talk a little bit about in the next lecture, has a means to switch the amounts, to modulate the amounts of these sigma factors. Yet another one for genes involved in chemotaxis, another one for genes at stationary phase, so genes that are necessary to survive in the stress of stationary phase, or genes that are involved in nitrogen metabolism. And you can see that the minus 10, minus 35 sequence changes tremendously, and sometimes it's not even minus 10 and minus 35. RPON uses sequences that are centered minus 12 and minus 24 away from the start of transcription site. So this helps the bacterium to dictate what genes are being transcribed at any one time. These sigma factors, even though there are several of them, there are ways that bacteria can increase the expression of the sigma factors at different times under different conditions. But bacteria also have the ability to respond more rapidly. So if a bacterium needed an alternative sigma factor for a particular environmental signal, if it had to transcribe that gene, that new sigma factor, and translate it, that takes time. And so if the bacterium is in a particularly stressful environment, the time that it took to make that new sigma factor might prove lethal. So many times what bacteria have done is that they have made a bunch of these sigma factors and they're holding them in reserve for just the right condition. So these are just two examples of sigma factors that bacteria use that they synthesize ahead of time. They synthesize when they don't need them, and they hold them in abeyance. They hold them in reserve with what are known as anti-sigma factors. So this is an anti-sigma factor strategy that's used by a soil bacterium, Clostridium. And this is actually the same genus of bacteria that's one of the predominant microbes in our intestinal tracts today, Clostridium. One of the ways that the bacteria makes certain that it only transcribes the genes to break down extracellular polysaccharides is that it only transcribes the genes to break down those polysaccharides when they're present. So Clostridia use this really clever system where they have a specific sigma factor bound basically and tied up in an unuseful form, bound to the membrane. So bound to this anti-sigma factor, which is bound to an extracellular receptor, this CBM protein for carbohydrate binding module, which basically is looking for extracellular carbohydrates. When they're not there, it binds to the sigma factor because you don't need to transcribe those genes because the carbohydrate is not there. But when those specific polysaccharides are there, that carbohydrate binding module recognizes those polysaccharides, that conformation change that information is transduced across the membrane, and the sigma factor is released. And so now that sigma factor can substitute into RNA polymerase, and so you can get transcription of the cellulosomal genes. These are the genes that make up the extracellular proteins that are going to be secreted into the extracellular environment to break down those polysaccharides, but only when the polysaccharides are present. When they're not there, this sigma is not available. It's tied up. And so when we come back on Friday, I'll give you one more example of another anti-sigma factor story that bacteria use. Otherwise, good luck on the exam this week. I'll see some of you tonight. I'll see all of you at some point this week. Hey!
Sorry, I just kind of popped up.
It's like the lights came on and there was somebody right there.
I do that.
This is